Hi everyone, it's an honor to share our work with you today. I'm going to introduce our work on rapidly studying COVID-19 virus polymerase complex structure using cryo-EM. We know that coronavirus are postal-sense single-strand RNA virus. After it invades and infects cells, it first directly uses its own positive-sense RNA genome to translate and synthesize RNA polymerase. Subsequently, RNA polymerase synthesizes antisense RNA with its genome as a template, and then transcribes message RNAs with this antisense genome RNA to obtain the expression of each coding protein. This antisense RNA is also used as a template to reproduce new genome and resemble new virus. This transcription and replication process is carried out by RNA polymerase. After years of careful study, number 12 non-structured protein were identified as RNA-dependent RNA polymerase, RDRP. Protein interaction network analysis showed that the coronavirus can assemble an efficient and somewhat profitable molecular machine with the RNA polymerase at the core and cofactor non-structured protein 7 and 8 as the primaries, and some other non-structured proteins to ensure an efficient transcription and replication of its large RNA genome with a length of about 30,000 base pairs. Interfering with activity of RNA polymerase, one of the most important antiviral targets at the heart of the transcription and replication machine, is expected to prevent the virus from replication. At the most recent ones, remdesivir and favipiravir are the products that target RNA polymerase with their metabolites. Cryo-EM analysis of polymerase and many nucleic acid binding protein will encounter the preferred specimen's orientation issue during vitrification. Usually, we have some ways to solve this problem, such as use fusion protein or antibody to alter the orientation distribution, or use a low concentration of detergent to change the surface tension, or use thin carbon film, commonly used for the large samples. Different sample preparation approaches, such as speed-up blotting-free vitrification, or collect image from tilled specimens. We have been studying some membrane protein complex structure for a long time, so we give preference to detergent solution, which give us the missing orientation projection finally. And the subsequent typical data processing process, accelerated by cross-spark, result in a high-quality reconstruction of 2.9 angstrom in 2 days. The 2.9 angstrom complex structure showed that the RNA polymerase of COVID-19 virus had the conserved typical viral RNA polymerase structure and an NI-RAN characterized domain of nidovirus. It includes typical palm, fingers, and thumb domain. The viral RNA polymerase NSP12 form a complex with one viral non-structured protein NSP7 and two NSP8. As early as 2010, it was found that the N-terminal domain of the SARS coronavirus RNA polymerase seems to be particularly important to the correct folding and activity of this enzyme. Defects in the N-terminal or the presence of artificially introduced redundant amino acid residues can significantly affect the stability and activity of the polymerase. Mutagenesis analysis show that its unique N-terminal NI-RINE domain has a profound effect on viral replication. What is exciting is that our structure completes this N-terminal domain information that has been missing in a previous research on SARS-CoV RNA polymerase. For the first time, a unique hairpin domain was revealed. We can see this hairpin interacts with both NI-RINE domain and the palm domain. What's more, it has been reported that zinc ions shows reversible inhibition effect on clonal replication in vitro in cell culture. We found that two typical zinc ion binding sites on this polymerase exhibit different zinc ion binding states under different purification conditions, which may provide a perspective for understanding the inhibitory effect of zinc ions. Meanwhile, by comparing with the polymerase HCV and the polyovirus, the typical template entry channel between the motif G and the motif F, nucleotide entry channel between motif E and the motif F, and the product release channel in COVID-19 virus polymerase can be found. Even more, by comparing with the complex structure of hepatitis C virus polymerase NS5B and the sofosbuvir, we propose a possible recognition mode of remdesivir. We also discuss some possible similarity and difference between the two. Similarly, favipiravir, another nucleotide analog, may work through a similar mechanism. If we have a little imagination and considering the possible paths and the moving direction of the template and the product RNA, we can provide an overall scenario in which coronavirus RNA synthesis is inhibited by nucleoside analogs. Of course, the real picture will involve the high-order structure of RNA and other members of the transcription replication complex, such as polymerase and hexase. There's a long way to go. In fact, since the outbreak of MERS-CoV, the coronavirus has changed from a newly emerging virus to a re-emerging and widespread virus. In the future, we may continue to face the threat of coronavirus for a long time. Fortunately, not only the efficiency of broad-spectrum polymerase antibody inhibitors show on COVID-19, but also the conservation of sp12 compared to the involving proteins such as spikes. They both suggest that polymerase is a promising and valuable target for broad-spectrum antiviral target drugs development. Despite some significant side effects, nucleotide analogs that target the polymerase may indeed be an effective drug candidate against infectious disease, particularly virus. Finally, I would like to pay an exceptional tribute to Shanghai Tech University and their administrative team, as well as the bioelectron microscopy facility for their great care and support to our research team to enable us carry on this research in a safe and healthy environment. It would have been impossible for us to get this work done without their tremendous efforts in the last 2 months during the COVID-19 pandemic. On behalf of our team, I also would like to convey our special thanks to Tsinghua University to their exceptional permission to allow students to go back to the laboratory to prepare the protein samples for this study. We also must express our gratitude to the campus service team of Shanghai Tech University Scientific Research Platform of Shanghai Institute for Advanced in Chemical Study, and National Center for Protein Science Shanghai, as well as all the manager and technician individuals who provide on-site or remote technical support. Their kind help and fearless support are pivotal to our work during the epidemic. We would like to thank the University of Queensland and Diamond Like Source for their collaboration. Thanks for your attention, and I would like to answer any question you have.
Thanks, Quan. And now over to Renhong.
Hello everyone, this is Renhong Yan from Westlake University. I'm the postdoc of Dr. Yi Gongshi and Dr. Qiang Zhou. Thanks the webinar organizers for giving me this opportunity to show our recent work about the structural basis for the recognition of the SARS-CoV-2 by full-length human ACE2. The outbreak of coronavirus disease 2019 has emerged as a severe pandemic. This disease is caused by SARS-CoV-2, a positive strain RNA virus. There are over 2 million people who have been infected by SARS-CoV-2, and over 100,000 people died from this disease. SARS-CoV-2, also called 2019 nCoV, is one kind of coronaviruses, which are important pathogens for human beings. There are 4 genera of coronaviruses. SARS-CoV-2 belongs to the betacoronavirus. SARS-CoV-2 is an enveloped virus with about 30 kilobase RNA, which can produce 9 major subgenomic RNAs. Among these viral proteins, the S protein on the viral surface mediates receptor recognition and membrane fusion. For the life cycle of human coronaviruses, these coronaviruses enter host cells by first binding to the respective cellular receptor through the S protein. Then the conformational change of S protein leads to virus and cell membrane fusion and viral entry. After entering into the host cells, the viral genomic RNA is released and translated into viral proteins. These proteins are further assembled in the ER and Golgi intermediate compartment to form a mature viral, then releasing from the host cells. For SARS-CoV-2, the sequence is highly similar to another coronavirus, SARS-CoV, which has caused severe acute respiratory syndrome in 2003 in China. So the scientists wonder whether SARS-CoV-2 and SARS-CoV share the same cellular receptor, ACE2. Then 2 independent groups used the viral infection assay suggest that ACE2 is the cellular receptor of SARS-CoV and SARS-CoV-2. The crystal structure of SARS-CoV receptor binding domain in complex with peptidase domain of ACE2 has been solved in 2005. Before we did this project, all the structures of ACE2 only contain peptidase domain. That of the full-length protein remains unknown. The full name of ACE2 is called Angiotensin-converting enzyme 2. ACE2 is a single transmembrane protein which contains an N-terminal peptidase domain and a C-terminal Collectrin-like domain. The main function of ACE2 is help the maturation of angiotensin, a peptide hormone that controls vasoconstriction and blood pressure. Besides this, the S protein of SARS-CoV and SARS-CoV-2 can bind to the peptidase domain of ACE2. As for the Collectrin-like domain, previous studies showed that the cleavage of C-terminal of ACE2 can significantly enhance the viral infection. The single transmembrane helix of ACE2 makes it challenging to determine the structure of the full-length ACE2 protein. So we did literature research and tried to find a binding partner with ACE2, and we found that ACE2 also functions as the chaperone for membrane trafficking of the amino acid transporter beta-1. Beta-1 is also known as SLC6A19, which can transport neutral amino acid such as leucine in the kidney and the intestine. So we wonder that the structure of full-length ACE2 might be revealed in the presence of beta-1. Then we co-expressed the ACE2 and the beta-1 complex in 293 F cells and successfully purified the homogeneous complex. You can see that the behavior of this complex is quite good. And used the good behavior protein, we make the cryo-EM sample. You can see the particles is quite good, and the 2D classification shows a good feature for this membrane complex. However, the data processing for this complex is not easy to do. It took us a lot of time to solve the structure of ACE2 and the beta-1 complex, because when the outbreak of COVID-19, I was on my spring festival holiday at home. Then we realized the severity of this pandemic and go back to lab to speed up this project. After hard working, we successfully solved a 2.9 Armstrong structure of ACE2 and the beta-1 complex. The high resolution supported reliable model building. You can see this structure is very beautiful. The ACE2 and the beta-1 complex is assembled as a dimer of heterodimers, with the Collectrin-like domain of ACE2 mediating homodimerization. We can see the homodimerization entirely mediated by ACE2, which is sandwiched by beta-1. Between the peptidase domain and the transmembrane helix is a neck domain. Both the peptidase domain and the neck domain contribute to dimerization with multiple interactions by some residues of ACE2. During the data processing, we also found about 20% particles showed a different conformation at the peptidase domain of ACE2. We call it open conformation. The previous one is called closed conformation. We can see that the major difference between the open and the closed conformation of ACE2 is that the peptidase domain are separated away in the open conformation, and the closed conformation has a closed peptidase domain. You can see the S protein of SARS-CoV-2 contains 1,273 amino acids with a single transmembrane domain. The functional S protein is assembled as trimer. The receptor binding domain mediates the binding with peptidase domain of ACE2. The extracellular domain of S protein of SARS-CoV-2 has been solved, which is quite similar with the S protein of SARS-CoV. Then we bought some receptor binding domain protein from Biological company and incubated the receptor binding domain of S protein with ACE2 and beta-1 complex, and prepared the cryo-EM sample. After 2D classification, we found an extended density above ACE2. We speculated this density might belong to the receptor binding domain of S protein. The structure of the ternary complex was determined to an overall resolution of 2.9 Armstrong. However, the resolution for the ACE2 and beta-1 complex was higher than that for the receptor binding domain, because the receptor binding domain is at the periphery of the complex. So in order to improve the local resolution, focused refinement was applied. This allowed us to reach a resolution of 3.5 Armstrong for the receptor binding domain, supporting reliable model building and analysis of the interface. So, as we can see, each peptidase domain accommodates one receptor binding domain. An extended loop region of the receptor binding domain spans the arch-shaped alpha-1 helix of the peptidase domain of ACE2. It's just like a bridge. So when we join in the interfaces, we can see the interfaces are mainly mediated through polar interactions. There are multiple interfaces in this complex. When compared with the crystal structure of SARS-CoV receptor binding domain and the peptidase domain of ACE2, we found the overall interface is similar, with the number of sequence variations and the conformational deviations are found in the respective interfaces. To confirm our structure, we aligned our cryo-EM structure with the crystal structures of SARS-CoV-2 receptor binding domain in complex with peptidase domain of ACE2, and found the interfaces are almost identical among these structures. Maybe you remember that I have ever said that previous studies showed that cleavage of the C-terminal segments of the ACE2, especially residues 697 to 716 of ACE2 by proteins such as TMPRSS2, can enhance the S protein-driven viral entry. This region forms the third and fourth helix in the neck domain of ACE2 and involved in the dimeric interface of ACE2. You can see this. The presence of beta-1 might block the access of TMPRSS2 to the cutting site on ACE2. Based on these findings, we proposed a simple working model for SARS-CoV-2 infection. When ACE2 stays alone, the S protein binds to the peptidase domain of ACE2 and starts the infection process. The TMPRSS2 can cut the C-terminal of ACE2 to enhance viral entry. When ACE2 stays with beta-1, beta-1 might protect ACE2 from TMPRSS2 digesting, so beta-1 might play an important regulatory role for the viral infection process. So, what we can do with this complex structure? First, we can use this structure to do drug design to block the binding between receptor binding domain and ACE2, that's protecting host cells from viral infection. We can also use this structure to optimize the neutralizing antibodies. This structure can also help with the development of vaccine. Finally, I would like to thank Professor Yigong Shi and Dr. Qiang Zhou for the support and help. I would like to thank the hard working of our team members, the Westlake University cryo-EM Center and the Supercomputer Center. This work was supported by the National Natural Science Foundation of China, the SARS-CoV-2 Emergency Projects of Department of Science and Technology of Zhejiang Province, the Key Program of Zhejiang Province,